Welcome to my YouTube channel. Today we are going to talk about adenosine triphosphate, or also known as ATP. So, the body is a complex organism, and as such, it takes energy to maintain proper functioning. And adenosine triphosphate is the source of that energy for use and storage at cellular level. So we will see that most of the processes that take place inside our bodies, they utilize this energy. So this energy actually is stored in the form of ATP, which is a high energy compound. However, we have many high energy compounds, but in this video we will focus on ATP. So starting with the structure of ATP. Structure of ATP, we will see that it constitutes we will see that the structure of ATP constitutes three phosphate groups, an adenine group, as well as a ribose group. However, the order, I did not consider the order there. Actually, it should be three phosphate groups, a ribose group, then an adenine group. Yes. So let us look at the structure of ATP. It begins with a ribose group, However, my drawing cannot be quite accurate. An adenine group, a ribose group is bonded to an adenine group. Sorry for that. And then we have the double bond between, because we know that we have from organic chemistry, we have the hydrocarbon, and then the carbon should always be bonded to four bonds. Should always have four bonds. So that is bonded to an amino group. So actually, the combination of an adenine group and a ribose group, that combination is what gives the molecule of ATP actually the name adenosine, or the adenosine part, combination of adenine and a ribose group. So also on this side, the three phosphate groups is what gives this molecule the name triphosphate. Those oxygen with a negative charge can actually attach some polar molecules, so it can be this molecule to be able to attach to other polar environments, or in hydrophilic environments. So actually, we say that ATP actually is the energy currency of the cell, and that is actually due to the electronegative charges exerting a repelling force between the phosphoryl groups. So there is a high energy that is stored within those bonds. So as I said, that high energy that is stored there is due to the electronegative charges that exert a repelling force between those phosphoryl groups. So That is why we call this molecule the energy currency of the cell. That is why we call this molecule the energy currency of the cell. So these bonds, when they are broken, they release that significant amount of energy that is stored within them, as we will see in just a moment when we talk about the hydrolysis of ATP. As we will see in just a moment, ATP hydrolysis. It actually refers to the breakdown of ATP molecule. Breakdown of ATP molecule in the presence of water to form ADP plus inorganic phosphate, releasing negative 7.3 kilocalories per mole. I'm sorry for that. It should be negative 7.3 kilocalories per mole. So however, this reaction is powered by the group of enzymes that are called ATPases. Also, ATP can be broken down to adenosine monophosphate, AMP, plus what is called a pyrophosphate, plus which you can say this reaction releases the negative 10 kilocalories per mole of energy. So actually it's an hexagonic reaction. It releases energy, because we know that there are endagonic reactions. We also have hexagonic reactions, which the reactions which, when the bonds are broken, they release energy. However, ATP, as we said, that it is used to power a lot of processes inside our bodies. It also works in intracellular signaling. In which we will see in our future lectures. It helps in signal transduction pathways, whereby we can say that the ATP can actually can be converted to what is called cyclic AMP, which cyclic AMP can actually stimulate the group of enzymes called protein kinases. Also, ATP can work at a DNA. It can work in DNA synthesis, because that energy that is released is used to power these reactions. When it gets hydrolyzed, that energy is coupled to power those reactions that need that energy. Also, it works in neurotransmission, whereby we can say that it can be used to synthesize neurotransmitters, as well as anchoring them to as well as transporting those neurotransmitters into the vesicles, and then anchoring those vesicles to the membrane so that they can be exocytosized in the synaptic cleft in our brain, or in our neural cells. Yeah. So however, all those others, we will see them in our upcoming videos. Thank you.